Hello students, today we will learn about the very important topic of medical diagnosis, ABO and RH blood group. First of all, the composition of blood. Blood consists two very important parts, plasma and the cell components. Plasma include amino acids, nitrogenous waste material, nutrients, electrolytes, protein and gases. In protein, the important proteins are albumin, fibrinogen and globulin. Globulin protein basically may be responsible for the formation of immunoglobulin or antibodies. In formed elements or cell components, you can find out here erythrocyte or RBC, platelets and leukocytes. Leukocytes broadly classified into two categories, granulocyte and agranulocytes. In agranulocytes, you can find out the very important cell lymphocytes. Lymphocytes broadly classified into two categories, B lymphocytes and T lymphocytes. B lymphocytes responsible for the humoral immunity, antibody mediated immunity, and T cell responsible for the cell mediated immunity. How we can separate these two layers? For that purpose, by phlebotomy, we have to collect the blood from the patient body. Centrifuge at very high speed. After this, you will find two layers. Upper layer termed as plasma, 55%. Lower layer is a cell component, including mainly RBC, erythrocytes, platelets, thrombocytes, white blood cells or leukocytes. Main function of RBC is oxygen transmission, gas transmission, including both here we can say is that oxygen as well as carbon dioxide. Platelets mainly responsible for the blood clotting, and WBC mainly responsible for the immune capacity. What is phlebotomy? Phlebotomy defined as the use of sterilized needle to draw the blood from the veins of the patient or person. If we collect the blood from the vein, it's termed as venipuncture. Almost 5 to 10 ml collected blood will be used for the further analysis, including microbiology, biochemistry, pathology, etc. in the laboratory. Definition of ABO blood group. The term blood group referred to as a specific pattern of glycoprotein antigen present on the surface of RBC. Mainly two kinds of antigen here we need to discuss in ABO blood group, A antigen and B antigen. What is the difference between two? The amino acid sequence of A antigen differ than B antigen present on the RBC surface. Next, RH blood group. System for classification of blood group according to the presence and absence of RH factor, RH antigen on the surface of RBC. If RH antigen available on the surface of RBC, it is RH positive. If it is absent, RH negative. The abbreviation RH derived from blood of rhesus monkey that was used by the scientists for the discovery of RH blood group. And in the diagram you clearly observe along with A or B, if this antigen available on the RBC surface, blood is RH positive. It is absent, blood is RH negative. Called Landsteiner, scientist who was discovered the ABO blood group, and RH blood groups were reported by called Landsteiner and A.S. Wiener around 1940s. In this slide, you can observe the four types of blood group under the ABO blood group system. The first one is type A blood group. In this blood group, you can observe A antigen on the surface of RBC, and in plasma, B antibody is available. Type B blood group, in which B antigen available on the RBC surface and A antibody available in the blood plasma. Type AB blood group, both A and B antigen available on the RBC surface, no antibody in the blood plasma. Type O, no A and no B antigen available on the surface of RBC, but both antibody available in the blood plasma of person. The person with O blood group considered as a universal donor, and person with AB blood group considered as a universal recipient. By the end of this class, we can understand the basics of experiment of blood group testing. For that purpose, we need monoclonal antibodies. Monoclonal antibody A, B, D. D is specific for RH antigen. Monoclonal antibody produced in the industry by using hybridoma technology. In three different vials, you can observe monoclonal antibody A, monoclonal antibody B, and monoclonal antibody D. D is specific for the RH antibodies that will be bind on the RH antigen. How we can use these monoclonal antibody? At the first step of this experiment, we have to collect a 3 drop of blood from the person, place at 3 different part of the slide. Even we can mark them also like A, B and D. Step by step, we have to add 3 monoclonal antibodies. Like here we mentioned we are adding the antibody A, and you can see already we have added the antibody B and D. After this addition, we have to use separate needle for the mixing of antibody, monoclonal antibody with the blood, and keep the slide for a few minutes, and after this we can observe the results. Antigen antibody reaction termed as agglutination, and if you provide the proper time, this reaction takes place perfectly. Like if we describe the first result, in the first result you can clearly observe that A antibody react with the A antigen present on the surface. No reaction in the B point, but you observe the clear reactions of D antibody or RH antibody with the RH antigen. That's why the final blood group, declared blood group for the person is A positive. In second result, you can observe that B antibody react with B antigens, RH antibody react with RH antigen, blood group is B positive. In third result, you can observe that A antibody react with A antigens, B antibody react with B antigens, RH antibody react with RH antigens. The blood group will be AB positive. In last, you will not find any antigen antibody reaction. It means no antigen available on the surface of RBC. This is a clear indication this is O negative blood group, or mainly O negative blood group termed as universal donor. Hope you understand this topic. Thank you very much.